Hello, my name is Kitty Deck. I'm a senior biology major at John Brown University, working in Dr. Wang's lab. We have been looking at the stress modulation of the antidepressant effect of ketamine mediated by ventral hippocampal parvalbumin interneurons in mice. To begin with, we are interested in ketamine as it is a fast-acting and long-lasting antidepressant that has been known to be effective in individuals with major depressive disorder who are resistant to other treatments. In looking at its effects, it is known to work through NMDAR dependent and independent mechanisms, which links ketamine to parvalbumin-expressing GABAergic interneurons or PV interneurons. We decided to focus on the ventral CA1 region of the hippocampus based on literature studies and previous work in our lab. Overall, we wanted to know how stress through unpredictable chronic stress or forced swim tests modulates the antidepressant effect of ketamine by ventral hippocampal PV interneurons and perineuronal nets in mice. For our methods, we use 36 male mice divided up into two groups. The first group is mild stress for 10 days, where they experience social isolation. The second group was unpredictable chronic stress for 4 weeks, where the mice were subjected to random daily stressors, such as tail suspension for 6 minutes, food and water deprivation overnight, or other stressors. After undergoing these stress protocols, the mice either received vehicle or ketamine injection, and then some of these mice also underwent forced swim test 1 hour after their injection. Next, brain slices were collected and they were stained for parvalbumin or Visia villosa L-glutenin. First, in looking at our results, this is the forced swim test immobility time. Immobility time will be reduced with an antidepressant. So in blue is vehicle, in green is ketamine. This is the mild stress group versus the unpredictable chronic stress group, where we saw no significance. This is consistent with literature. Next is the results of our staining, looking at the number of PV positive cells and the number of VVA positive cells. Here, the yellow bars represent mice who did not undergo forced swim test but received vehicle. Red bars did not undergo forced swim test either, but they received ketamine. Blue underwent forced swim tests and received vehicle, and finally green underwent forced swim tests and received ketamine. Here we see several areas of significance. First, with the number of PV positive cells, we only saw significance in the mice who experienced the unpredictable chronic stress. Here we saw a smaller number of PV positive cells in mice after forced swim tests compared to those who did not experience forced swim tests for those who received the vehicle injection, seen in the yellow and blue bars. We also saw a larger number of PV positive interneurons when mice received a ketamine injection prior to forced swim test. Next, with the number of VVA positive cells, we saw a stress effect with the yellow and green bars, where there was a larger number of VVA cells associated with UCS in mice who experienced no forced swim tests and vehicle injection or forced swim tests and a ketamine injection. Also with mild stress, we saw a smaller number of VVA positive cells in the mice who had a ketamine injection after forced swim test, and we did not see this in the UCS group. In conclusion, with PV interneurons, if ketamine is administered before forced swim tests, it can rescue PV positive cell number in mice who experience UCS. As for perineuronal nets, which is a specialized extracellular matrix of which VVA is a protein within it, these are affected by long-term and short-term changes with stress and ketamine administration. In the future, we would like to expand this research and investigate other regions and compare changes seen there due to ketamine, UCS, or FST. I would like to acknowledge Dr. Wong's lab and all of the work they've put into this research, and you can find our contact information here as well. Thank you for listening.